TGF beta signaling pathway. TGF beta transforming growth factor beta signaling pathway is a key regulatory pathway involved in the controlling cell growth, differentiation, cell death, and immune response. It is important for development, wound healing, fibrosis, and cancer progression. So, two main TGF beta signaling pathways are there, canonical and non-canonical. The canonical is SMAD dependent pathway, and this is a classical and most studied TGF pathway. There are various stages in this canonical pathway. First is the ligand binding, where the TGF beta ligand binds to the TGF beta 2 receptors 2, and the TGF2 recruits a phosphorylates TGF type 1 receptor, and then SMAD is activated. TGF beta 1 phosphorylate receptor regulated SMAD, R-SMAD, which then activate the SMAD2 and SMAD3. Then the complexes form, phosphorylated R-SMAD form a complex with SMAD4, that is co-SMAD. Then nuclear translocation is done. The SMAD complex enter the nucleus and it binds to the DNA and interact with the transcription factor to regulate the targeted gene expression. The non-canonical SMAD is SMAD independent pathway, and these pathway bypass the SMAD protein and use other signaling cascade. For example, the MAP, the PI3K and ATK pathway, the ROC like GTPase pathway, and NF and KB pathway. These regulate the cytoskeleton organization of the cell survival and migration. SMAD7, an inhibitory or SMAD or iSMAD, is induced by TGF beta itself, a negative feedback loop. The SMAD7 blocks the R-SMAD phosphorylation and recruits ubiquitin ligase, that is MRF1/2, to degrade the receptor. So it is the regulation and termination of the signaling. So TGF beta receptor are serine and threonine kinase and not tyrosine kinase. The SMAD2 and 3 will activate the R-SMAD, and SMAD4 is the activate the co-SMAD, and SMAD6 and 7 are the iSMADs. So SMAD complex function as a transcription factor. Phosphorylation of R-SMAD at C-terminal SS axis motif is essential for activation, and the pathway regulates both cell cycle arrest and cell differentiation depending on the cellular context. Thanks for watching. Do like, share and subscribe.